So now I will tell you how we can use lifetime measurements to look at molecular binding. And so here is a challenge that we typically encounter in biological research. Let's say we have a protein A and we have a protein B, and we want to know if these two proteins bind. So many physiological processes require these events to take place, happen all the time. You could have two proteins binding, but this could also be a drug targeting a certain protein, and you want to know whether it does that inside the cell. How can we detect this? I mean, one of the things that we already know is we could put a green fluorophore, let's say, on one protein A, and we could put a red fluorophore on protein B, and then we could just look at the two colors and then just say, okay, this is how far they are apart. But of course, when they get together and bind, these distances are very small. They are, of course, of the order of the size of a protein. Now, the problem here is that if you have protein A, shown here in this cartoon, if that's the actual physical size of the protein, so this is drawn to scale, then its point spread function looks something like that on the detector. And protein B, conversely, I mean, of course it's red, I can distinguish them, but I can only distinguish them if they are further apart than the point spread function. If they are less than the width of a point spread function, these blobs are just indistinguishable. We don't know, we can't say whether they're 100 nanometers apart or whether they're 10 nanometers apart or 1 nanometer apart. They just appear to us to be in the same position. So to overcome this problem, there's a technique called Förster resonance energy transfer, or FRET for short. And so this is how it works. So if you have the green protein A, and the other one is shown here in yellow, it was shown in red beforehand, but let's suppose I have a blue photon, I excite the green fluorophore with it, then I get signal from protein A. Protein A is called the donor molecule. And so if the molecules are very close together, they actually need to be within 5 nanometers or so, and certain conditions are met, then energy from the donor that I've excited can hop over to this other protein, the protein B in this case, and this protein is, for obvious reasons, called the acceptor. So if these conditions are met, and they only happen when these molecules are very, very close to each other, then although I've excited the donor molecule, I actually get a signal from the acceptor molecule. And so this process is shown here in an energy diagram as follows. Here's my donor molecule. I excite fluorescence up. I excite population up into its upper state. Of course, it can come back down by normal fluorescence processes, but if the conditions are right, energy can hop over to this other molecule, the acceptor molecule, and then this one can give us light back rather than the other way around, that the donor giving you emission. So FRET only occurs when a number of conditions are fulfilled, and I'll go into those in a second. One is that what you need to have is you need to have the donor emission spectrum, shown here by this green wing on the right. This donor emission spectrum, so the spectrum of the signal that comes from the donor if it's just excited on its own, that must overlap with the excitation spectrum of the acceptor. So in other words, the energy band of the photons that emit from the donor must overlap with the excitation spectrum of the acceptor. Okay, and then the acceptor emission spectrum is of course Stokes shifted to the right. So the whole process is, you know, the FRET photon that comes back is redshifted quite extremely right from the original donor. And so the next thing is that the molecules need to be oriented with respect to one another in the correct way. We say that the electric dipoles need to be aligned, more or less. And the reason for that you can understand. the Fret interaction, we don't have time here to really go into the detailed physics. It's quite complicated. It's quite beautiful physics, but it's too much to do in this course. But in fact, it turns out that this interaction is an electrostatic interaction. So you can think of the donor as emitting an electric field, and the acceptor feels that field, right? And it depends how the two things are oriented. Now, if you remember when we talked about these conjugated bonds in organic molecules, electrons moved along certain directions. And so the way that these are moving, of course, the way they can interact with other things now depends on their orientation. Like an antenna. If the antenna isn't oriented in the correct way, the electrons inside the antenna don't radiate energy very much, right? Okay, so the orientation must be right. And finally, and most importantly, the distance must be right. This process only occurs when the molecules are to within 5 or 10 nanometers from one another, or let's say less than 10 nanometers, definitely. Beyond that, the signal becomes so small that you can ignore it, and that's actually why it works. It reports only on these very short distances. As soon as the molecules come apart, the process doesn't take place. And that's fortuitous, in fact, that this happens over this length scale, because otherwise it wouldn't work as a molecular reporter. This is the man who came up with the theory. He wrote a beautiful paper about this in 1948, and he was a really sort of an unsung hero of his time. He really discovered a lot of stuff on the photophysics, also about photosynthesis and so on. And in fact, he was Theodor Förster was is behind this Fret theory as well. So I'm now turning to the paper copy again, the paper visualizer again, just to show you how this works. So let's go back to our two-level system. And now we draw in again. We say we have low light intensity and low population in the upper state, so we ignore stimulated emission. So we have our usual processes, A21 and Q21. And now Fret is now an extra loss channel. So this here is the donor molecule. The donor molecule, the donor fluorophore that's attached to the protein I'm interested in, one of the proteins that I'm interested in, if it's on its own, it will just emit light in the usual way and have a certain lifetime. But if another molecule is nearby, there can be another loss mechanism. And so the lifetime of the donor, T D in the presence of Fret, is equal to 1 over A21 plus Q21 plus K Fret. So there's this extra term in there now. And so what that means is that if Fret takes place, then I can easily read this off the fluorescence lifetime, because perhaps the original lifetime of the donor on its own is just this, tau D. That will be tau D. And then I put the other molecule very close by. And as soon as that happens, okay, now this one can actually also lose energy over to that one. So it's like putting another hole into a bucket, so it empties much more quickly. But if you put several holes in, it leaks its content more quickly than in other ways. So the same way here, by having the other one there, it can actually leak energy not just by Q and A21 and Q21, but also by K Fret. And so its excited state population will decay more quickly as a result. And so here is the same thing when you have Fret occurring. So this will be TD Fret in the presence of Fret. And by just comparing those two things, you can say whether something is taking place. So the rate K Fret at which this happens is equal to 1 over tau D. So that's essentially remember, tau D is just 1 over A21 plus Q21 without Fret. And now there's a factor in there, which is R0 over R. And here comes the important part, to the power of 6. Now, R0 here is the Förster radius. And it is typically 5 to 10 nanometers for fluorophores that you use in biology. And the efficiency of transfer is given by K fret. So it's essentially, what's the efficiency of transferring energy across to the other molecule? Maybe what I should do is I should draw what happens. So let's suppose I have a ground state for the donor molecule and the upper states for the donor molecule. I excite population up like so, and then of course we can have A21 and Q21 taking place as usual, right? But now I have overlapping energy levels of an acceptor molecule. So this is the acceptor here, and over here this is the donor. And of course, if this is given, then I can have transfer at rate K fret, and then this one can come down. I can, and this is the acceptor fluorescence. Okay, so the efficiency of the fret process is given by comparing how much goes across in terms of energy over the total loss of energy from the donor. So how much of the total energy that you have in the donor is actually going in there. So I divide this through all the other rates. So that would be A21 plus Q21 plus K fret. So these are all loss rates. So it's the energy transfer rate divided by all loss rates. And that turns out to be just 1 minus T of the donor in the presence of fret. And of course, this comes straight from these equations. You can just test that yourself. You see it immediately. So simply by comparing the donor lifetime on its own with the donor lifetime when fret takes place, I can work out the efficiency. And it turns out that this efficiency at E equals 50% when R equals R0, i.e. when the donor and the acceptor are within 1 Förster distance. So if the Förster distance is 5 nanometers, if the donor and acceptor come to within 5 nanometers, then 50% will go over to the acceptor, and 50% of the donor energy will just be lost in the usual way, right? Within the donor itself, right? And so this is remarkably interesting now, because I want you to work out what happens if you double the distance. Because of this r to the -6 dependence of the distance, of course the signal very rapidly goes to 0. And so that's why this technique is in fact so useful. And it's also called sort of a molecular ruler. So I will go and show you now back on the computer a few more things. So you can see why fret is useful in the following examples that I'm going to give you. So fret only occurs if the dimolecules are in close contact. So if they are within less than 10 nanometers, you can be pretty certain that they have bound to each other, because it's unlikely, right? Because if they just pass each other, this passage will be too fleeting, and so on average they'll be too far apart for fret to occur, to be detectable, right? So only if they really are bound would they be closer than that distance. And so you can make now sensors, you can measure interactions between proteins or between proteins and drugs and so on, simply by putting a donor and an acceptor on the specific molecules. And you could in principle even measure distances, because the efficiency will change as a function of distance between these two. Although because of this nonlinear dependence, it's actually very hard to measure a range of distances, but it can be done for certain systems, and I will show you one of those. So how do we measure fret? We excite the donor, and we look at the emission of the acceptor. This is called sensitized emission, or we actually do what we did before. The more sensible thing is to measure the fluorescence lifetimes. So here I show you one example of how we used FRET in our own research. This is a while ago, to look at the interaction of two proteins that orchestrate cell division. So these are cartoons of what these proteins look like. And so at the end you see these yellow tails, and these are a donor molecule that we've attached to the proteins using this fusion protein technology that I've already talked about. And so we have a donor on this molecule and the acceptor on the other molecule. And then inside the cell, these come together, and you will see how the FRET signal increases as more and more of the molecules bind, and then at the peak the cell will start to divide, and at that point the signal goes down. And so I don't want to give you all the details, it's too much, but you can see that you can get molecular level information. So here's an image that shows a cell that is just about to divide, and what you see is the FRET signal that is from these two proteins, cyclin and CDK they're called. And if I switch on the camera, the color of this turns more and more red, and what that means is that the FRET signal gets stronger and stronger and stronger in time until it reaches a maximum about now. Now the switch falls apart, and just at that point where this switch falls apart, you can see the two daughter cells forming. So this FRET signal is coming together of these two molecules, is tightly orchestrating the cell division process itself. So this was done by looking at the emission, just looking at the intensity ratio, how much light comes from the donor, how much light comes from the acceptor, and ratioing them. So this is shown in a cartoon way down here. Don't worry about the details. This diagram down there, you don't need to remember. But the point is just this: by looking at the intensities of the two things, we can ratio them, and we can say how the FRET level increases. So as the acceptor emission rises and the donor emission goes down, ratioing those two things and calibrating our instrument correctly, we can actually quantify the signal from these two molecules. And there are other things you can do. You can make environmental sensors. So this here is called a chameleon sensor. So essentially what you do is you take a donor molecule, a fusion protein again, and an acceptor molecule, and you fuse it to a sequence, a protein sequence that is responsive to the presence of calcium ions. So when calcium ions are present, this middle part here is engineered to act like a hinge, right? So when the calcium comes, this hinge sort of closes, and the donor and acceptor come close together, and you suddenly get a strong FRET signal. And what that allows you to do is, so you see that as you change calcium concentration, the donor lifetime goes down, indicating more and more FRET, because the hinges is locking together. And so we can use this, in fact, to measure KDs, or so-called dissociation constants. So we can actually measure binding strengths. And you can do that by, so essentially this is an equilibrium constant of the unbound species, the ratio of the unbound species, the concentration of the unbound species or the unbound states compared to the bound states, right? And so so at any one point, you get then a population of FRET signals. I mean, so in the unbound state, you will have the donor lifetime is unaffected. So the fraction of molecules that are not interacting with the calcium will have a decay curve like this, and the fraction that have bound calcium will actually hinge together and give you FRET. And from that, so you can get a dissociation constant. And in fact, there's an exercise in the question sheets that you will get where you can see how this works in practice. Okay, And so finally, I can show you how people have in fact used FRET also to measure distances. So here they've just attached donors and acceptors at predefined positions along DNA strands, and you can see how really the FRET signal behaves, the FRET efficiency behaves, as we've seen from the theoretical calculations, the considerations that we had beforehand. So for these systems, it provides this information. You can also use it here to measure molecular dynamics information. So for example, people have looked have designed these little beacons here. So strands of DNA sort of that fold together, and then they they open and close and open and close, right? And so again, the equilibrium dynamics. And when they are open, you get a strong donor signal and a low acceptor signal, and when they're closed, the opposite takes place. You can see they are closed, open, and so on. So strong FRET, low FRET, strong FRET, and so on. So it's remarkable that you can get sort of dynamic information like that on the nanoscopic scale. So I should conclude that what FRET does is it gives you information at very small, much smaller scales than the wavelengths of light, but it does so indirectly. It does it by looking at spectroscopic information, i.e. the lifetime in this case, rather than actually producing images that are better resolved. I mean, the images you get with these methods, I mean, if you look at the FRET image I showed you of the cyclin CDK, you're getting information about molecular scale processes, the cyclin and the CDK, the two proteins coming together causing cell division. But of course what you don't do is you don't see the detail at the 5 nanometer level. You only infer it from the information that comes back from your signals. And so the actual images you produce are not better than diffraction limited. They are just diffraction limited in the normal sense, lambda over 2Na. But what I will do in the final lectures, I will go and talk about techniques for which the Nobel Prize was awarded to Betzig, Myrna, and Hell, namely optical super resolution methods, where in fact the images themselves are more highly resolved than what you'd get by normal Abbe theory and diffraction limited optical imaging. And we'll talk about these from the next lecture on. Thank you.